So yeah, I'm going to talk about the long-read nanopore sequencing, whether this can be a one-test-fits-all in our laboratory. So a little bit about our route towards application in a clinical context. So a disclosure slide. So our lab, so we're located in Utrecht in the Netherlands, and so we're one of the 8 diagnostic centers in the Netherlands, and we do about 90,000 tests a year. We've been an early adopter of next generation sequencing, and actually, so last year only we started with the short read whole genomes, and actually beginning of this year we put the exomes to rest and fully switched to short read whole genomes, but perhaps those are kind of short-lived. Perhaps next year we'll transition to the long read genomes. So long read whole genome sequencing can this be a generic technology? So in the past we've been using many different technologies to try to find all the variants that are relevant for us. So yeah, we hope that this long-read sequencing can be one test fits all and reduce this diagnostic odyssey for patients to ultimately also make it cheaper, quicker, easier, so everybody's happy. So this is the workflow we developed. So we do the sample prep on the Callisto from Volta Labs. It can do 24 samples in one go. And it's a plug-and-play kit that we use. We use the P24 for sequencing. So we made a different decision. So we didn't want to wash and reload, and so we decided to go for 2 flow cells per patient, and also a little bit anticipating improvements in the flow cells, so that later on we can maybe go back to 1 flow cell again. So we do runs with 24 flow cells together. We also do machine base calling and mapping, and we use the HEC version 5.2.0, and we do the methylation in all contexts. And then our third module, the bioinformatic analysis. So we basically use Epi2Me, the human workflow, out of the box. Didn't do any optimizations on that, apart from merging the data of the 2 flow cells. And then for the last module, we intend, we didn't have much experience yet, but to push this towards our tertiary analysis platform, Franklin. So the first thing we did is to look at what kind of variants are we finding today with our short read exomes, short read whole genomes. And it's about like 75% of the variants are SNPs and indels. Then about 20% of the variants that we report are CNVs. Then some like 5% STRs, and a very little amount of cases with aberrant methylation and other complex variants like inversions. And so the samples we put into this study are all archival material where we did find previous variants, and we just wanted to stress the system. So can we find these back? And we want also to have a sort of a reflection of what we find in general in our general workflows. So this is the sample selection we did in this study. So in total we selected 123 diagnostic samples, and which comprise in total 166 diagnostic variants. And we also included genome in a bottle for extended SNV and indel validation. So in the end, we put in more CNVs than SNVs because we use also this genome in a bottle for the SNVs and the indel. So in total, there were 66 CNVs, 44 SNPs and indels. Also a little bit of focus on the STR, so 36 of these across different genes. We put in 7 homopolymers, 7 cases with methylation, and also 5 cases with inversions. So this is the workflow we have. So we use the fast prep for our DNA fragmentation, and then the Callisto for the library prep. So then so there is enough of this library prep to split it and to load 2 Promethean flow cells. So that's exactly the same prep that goes over 2 flow cells. And that gave us also some insights that there is quite a lot of flow cell variability. Sometimes you have a flow cell that really outputs just like 30 gigs or something, and the other one is outputting more than 100. So that was also a surprise to us. So we do 72-hour runs without washing and reloading. Then we do on-machine base calling mapping, and then we merge the data from the 2 flow cells and run the epitomy variation workflow. And in the end, we intend to upload it to this analysis platform. Some short QC metrics in our validation cohort. So this is per flow cell. So you can see there is quite a variable output of the flow cells, but the mean is about 90 gigs per flow cell. Also our N50, the mean for this is 14, but also quite variable. We're using archival DNA, and sometimes, they're just a little bit worse than others. And we did see a pretty strong correlation between sample age, sort of. So the more sort of fresh samples, they work better than older samples. So this is per sample. So we reached a mean genome coverage of almost 60x, and we had only 1 sample that did not reach our 30x threshold. Our DNA length, it's basically the same as what I said before. So the mean is 14 kb. So first of all, then the genome in a bottle validation. So I compared this with our current short-read whole genome sequencing done on Illumina. These are both samples with a coverage of about 38x, and you can see that the recall is really similar between the 2 technologies. The precision maybe for the indels is a little bit higher for the Illumina, but it's all within basically also the boundaries we set for our diagnostics. So this really looks great. So what about our real pathogenic variants? So we put in 51 of those, and this is the difference between all the variants. So we had 21 SNPs, 7 small deletions. We had 7 homopolymers. We had 6 delins, 5 little dops, and 5 small insertions. And we found them back, all of them. So, and even most of these variants were nicely phased. It's very clean data, and our conclusion is, this is really suitable for diagnostics. And this is a little example of one of the homopolymers we tested, and it looks great, and I think there will be no big issues on this. We didn't stress the homopolymers. We looked at what do we find in normal diagnostics, so that's kind of up to 10 bases of homopolymer. How it performs even longer, we don't know yet. But we don't really find them in our current diagnostics anyhow. So. Then next, the CMV validation. So in total, these are 66 variants. And on the left, you see a graph of the size distribution of CMVs we currently find in our lab. And I want to stress that we find a lot of small stuff. I think in many cases, these have been overlooked. We have a really long history in looking at the CMVs, from exomes, but also from the short-read whole genomes. And it's a quite big proportion, the really small stuff. So we try to also reflect that in this validation study to put in the challenging CMVs as well. So we put in quite a lot of small ones, which are smaller than 100 kb. So the variant calling we did, so it's based on the Human Variation Workflow version 2.7.2. And there are 2 variant callers for this. There is the ONT Spectre variant caller, and this is designed to detect larger CMVs above 100 kb. and this is based on coverage. And we have sniffles. It's a structural variant caller designed for long-read sequencing, and this is really sort of read-based, so it's looking at the clip basis, and etc. Keep in mind also that 45% of our CNVs in the study are below this 100 kb threshold, so perhaps Spectre is kind of not suitable for those. So we only have sniffles for these variants. So these are the results. So I split them between the different variant callers. So if you would only look at Spectre, we have a recall of 34 variants of the total of 66. So we have a recall of 51%. If you only look at sniffles, we can call 27 of the total of 66 variants. So that's a recall of about 41%. If you are only looking at the variants that are called by both callers, there are actually only 8. So, yeah, they work in different fields, sort of these variant callers. But when you combine them and you, whether they're called either by sniffles or by Spectre, we find back 80%. But we're still missing like 13 variants. But conclusion about this is that you really need both of these callers to detect the full scope of CNVs. So why did we miss so many of these CNVs? So one reason is that in this standard workflow, the CNVs in the PAR regions on the X chromosome are blacklisted. So you will never be able to find them if you blacklist, of course. So we had, I think, 3 or 4 of those in the set. We also saw that there are quite a lot of CNVs in regions with high coverage noise. So the coverage there is quite variable. And if you are leaning on the coverage-based caller, yeah, it might be difficult to call. The start and stops of some CNVs were not correct. We had a big, high mosaic full X chromosome deletion that was not called. Yeah, it was not in the VCF. It was sort of called in the QC metric, but yeah, we really wanted it in the VCF. So, and what stood out a little bit is that there might be a group of tandem duplications, especially, that are a little bit longer than your read length, but they're smaller than this 100 kb, and they might fall in a sort of dark region between the two variant callers. So we looked a little bit more in detail. So this is an example of how the noise looks in the sniffles analysis. So the top graph here is a normalized coverage that comes out of the sniffles pipeline, and you can see it's kind of weaving, right? And here is the duplication, and it's sort of within this noise. And I can, well, visually, I would not be able to detect it. So yeah, how would the software do that? So we tried another variant caller. It's called QDNA-seq, and it does a GC bias correction. And there you can see now really nicely that this variant is clearly visible, and this was actually also called by QDNA-seq. And what's also nice is that this duplication is flanked by LCRs. So it will also the SV caller, yeah, it won't find any informative reads to call this. Another example, again, really a lot of noise in the sniffles data, but if you normalize for the GC bias, you can really quickly see it again. We looked a little bit deeper in this GC bias, and we think it's part of the reason why some of these variants are not called. It also causes incorrect breakpoints. It introduces a lot of false positives. And so we started to look at it a little bit, and we so there is no GC, QC anywhere in the ONT workflow. So we developed a little bit sort of a mean deviation score. It's sort of a proxy for GC bias. And we plotted that towards the total CNV size we have in our data, and also the number of CNV calls. And there is really, really strong correlation. So yeah, this GC bias is variable. and we see that it's correlated with a lot of increase in false positive calls. These are really, very difficult if you have these in your diagnostic workflow. And these are not small; they're like 10 MB false positive calls. And I hope I can convince you with this plot is that this is chromosome 1 in a sample, and this is the output of sniffles. Actually, this is a false positive call based on the sniffles data. And when you correct it with qDNA-seq, you can see it's a really tight, nice ratio plot that I think we can all appreciate will work well with CNV calling. So yeah, that's what we did. So we looked at this qDNA-seq CNV caller. So it's an older variant caller for these CNVs based on coverage. It actually does include this GC bias correction. We think it's really important. So the PAR regions on the X chromosome are not standardly blacklisted. And you are flexible in the target size, so you can set it to like 5 kb instead of the 35 kb, I think it is, for the sniffle. So you have a higher resolution, more points to, more data to calculate. So this finds actually 10 out of the 13 missed CNVs. So we're really happy with that. It's actually also part of the human workflow, but you have to activate it again if you want to use it. So what about these other 3 missed CNVs? So this is an example of a tandem duplication of 14 kb. And you can see this also in this coverage plot, but you can also see here nice clipped reads. It's tandem duplication of 4 exons in the TLK2 gene. Pathogenic, explained the disease in the child. But yeah, it was not called in the workflow because it is of course below, and also so the qDNA-seq didn't call it. Probably it's for that caller as well under the threshold to call. So you're stuck with the SV caller. But yeah, we don't know yet why this is not called. So our conclusions. So I think the positive news is, of course, it's all in the data. So we can see all the CNVs, and it's just better to tweak the tools to get them out of the data. And currently we have a 95% recall rate. So we need GC bias correction. I think we as a user should be able to easily configure these blacklisted regions. And we need some improvements in the CNV and SV callers. Also, we need more QCs. So we want to have more sample QCs. So, for example, a metric for GC bias, but also once the variants are called. So not all variants are equal, right? Some of them are a little bit less quality and stuff, and we need to be able to tweeze that out and to be able to filter on it, and so put the user in the power, basically. And we need visualization. So there are standardly no visualization present. So you need these SNP array-like ratios to evaluate the CNVs, to have the correct breakpoints. We also need B-allele frequency plots, which also help you to redefine these boundaries. So that's one thing we're actively working on with the ONT team to improve the CNV workflow. So the last part of my presentation, it's about the short tandem repeats. So the standard tool in the human workflow is Straggler at the moment. And these are the results. So we have multiple genes plotted in one graph with their repeats. These are in the normal range, but you have also the expanded ones. And the nice thing is that the estimation of the real pathogenic repeat length, this is working really a lot better with the ONT sequencing compared to our PCR results. With PCR, you always have this upper limit, and beyond that, you can't really determine the size anymore. What's a little bit disappointing is that there was one C9ORF72 expansion missed. It was this one. And that's probably because straggler needs real repeat spanning reads. And as soon as your repeat is longer than your read length, straggler won't be able to call it anymore. And we also see some small discrepancies in the normal range. So in parallel, we started testing other tools. So one of them is Abacus. You can find it on GitHub. It's been developed, I think, in Denmark. So this gives also a better estimation of the complete repeat expansion. So that's very similar to what we see with straggler. All expansions are correctly called, and we also have a very good correlation in this normal range, which is really small things. Things keep progressing. So we also tested an in development STR caller, and actually this one performs even better than this Abacus, although the differences are really small. But we're really confident that that this repeat expansion testing, this is really gonna fly with the ONT sequencing. It works really well. And of course, for example, for the FMR1, you can also interrogate the methylation, and that also looked really nice for the full expansions. Our conclusions. We developed a high throughput ONT sequencing workflow that basically works in most cases and on most samples. The sensitivity for the diagnostically relevant SNPs and indels is greater than 99%. For the CNVs, it's about 95%, and for the diagnostically relevant repeat expansions, this was also 100%. We're really confident that this is a big step towards clinical implementation, although there is a little work to be done, but we're working with ONT to especially improve this the CNV workflow and also the visualization. So with that, thanks to all the people who contributed to this work, and hope to discuss this with you guys.